So this next section talks about organic and biochemistry. To begin, we're going to define what organic is. Organic is not talking about don't use pesticides in your food. It is talking about a compound that contains carbon and hydrogen. So organic molecules contain carbon and hydrogen. Inorganic do not contain carbon or hydrogen. Carbon and hydrogen. So they could contain hydrogen, but not carbon. So anytime you have carbon in something, that's organic, and that's involved in life. And since we're talking about anatomy and physiology, organic compounds are a major thing about how our body works. It's a major component of our structure, our metabolism, and our lives. There are many organic molecules that make us function. I'm just going to focus on the major ones. So the first one is a ketone group. If you've ever heard of diabetic ketoacidosis, look at what this ketone group looks like. So it's this carbon, double bonded oxygen, and then you have two random side groups. So random side group 1, random side group 2, and then you have this double bonded oxygen here. In ketoacidosis, ketone bodies are produced because the patient is using fats for energy production instead of using sugar for energy production. Why is this happening? Well, diabetics, they don't have the capability of processing sugars like people that are not diabetics. So if they aren't taking their insulin properly, they aren't following their appropriate guidelines in order to moderate that condition, then with ketones, they get produced. They can produce acids. The patient slips into a coma, get all delirious. You may smell acetone on their breath or alcohol on their breath, and there's a lot of bad outcomes from that. Ketoacidosis is something you'll explore in detail once you get to advanced A&P. For this class, you just need to know what a ketone group looks like, and it's there highlighted in red for you. An alcohol group. An alcohol group has an OH on it, so that's alcohol. That OH group allows for increased permeability in the body, so it can go through cell membranes a little bit easier. It can get through the blood-brain barrier definitely, and that's why when we consume alcohol, we end up getting drunk from consuming alcohol. So here we have this OH group, and that makes it pretty soluble if a substance wants to travel through a membrane. Macromolecules. Macromolecules are large molecules. So macro means large or pretty huge. So with the macromolecules in the body, those are discussed on the next slide here. Carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates are sugars. Lipids are fats. Proteins, we just call those proteins. And nucleic acids help make up DNA and RNA. So now we're going to delve into each one of these in a little more detail. Starting with the carbohydrates, the sugars. We have monosaccharides, disaccharides, and polysaccharides. Monosaccharides is talking about one. So monosaccharides are the building blocks to all carbohydrates. It's one individual sugar unit. So one is a monosaccharide. It's one individual sugar unit. So then you can deduce the definition of a disaccharide if you know what di means. So di means two. Disaccharide consists of two monosaccharides that are bonded together. So an example of a monosaccharide would be glucose, galactose, or fructose, while an example of a disaccharide would be something like sucrose, maltose, and lactose. Sucrose is actually glucose and galactose bonded together. Maltose is two glucoses bonded together. And lactose is glucose and fructose bonded together. And that's how we get these disaccharides. Now polysaccharides, poly means many. So polysaccharide contains many monosaccharides or many building blocks. Examples of polysaccharides include glycogen, which you would predominantly find in animals. Our muscles store sugar as glycogen. Glycogen is kind of the basis of liver storage of glucose as well. And then we have starch. And starch is going to be the polysaccharide that's found in plant material. There's a bunch of other polysaccharides. Cellulose is one of them. Cellulose forms part of the outer cell wall of plant cells, and when you have cellulose abundant in a plant, people don't have the capability of breaking down this cellulose, so this is why when you eat corn, you end up pooping out the corn, because we don't have the enzymes that are necessary in order to digest cellulose. Actually, no animal on earth has that capability. It's all dependent on the bacteria that's found inside of that animal that could break down cellulose. So if it's an animal like an herbivore, like a cow or something, where cows eat a lot of grain, the cow would never be able to get nutrients from its food without that bacteria being present inside of its gut. Now moving on to the fats, so the lipids. Okay, so triglycerides. Tri means three, and you can see the structure of the triglyceride here. This is the major storage form of fats that you're going to find in the body. Here you can see the glycerol component of the triglyceride. So we have glycerol, and then we have three fatty acid chains. So the building blocks of lipids include glycerol and fatty acids. So now we'll talk about the difference between saturated and unsaturated fats. Saturated fats are called saturated because they're completely filled up with all of the hydrogens that this molecule could possibly hold. Every carbon is forming the maximum of 4 bonds that it could potentially form. Saturated fats are abundant in animals. And these are solid at room temperature. So butter that came from milk from a cow, that would be a saturated fat. Unsaturated fats do not hold all of the hydrogens that they possibly could. So these two are missing hydrogens. Now when they miss hydrogens, they end up forming a double bond. And anytime that you have a double bond, it forms a kink in the chain. And because of these kinks, these can't pack into a solid at room temperature. So unsaturated fats are typically liquid at room temperature. These are going to be all of your plant-based oils. So unsaturated fats are predominantly found in plants. So what is a trans fat? Sure, you've read labels that say no trans fats and so forth. Well, what a trans fat is they took an unsaturated fat and they took this cis double bond and turned it into a trans double bond. This was done in a laboratory. This is not done in nature. So because this was chemically modified in a laboratory setting, a lot of people say that this is bad because it's been modified. Well, this is still a fat. You're still able to break it down. You still get calories from this fat. But other people say, oh, well, because it came from a plant, it has to be healthier for you. So they assume that it's less calories that you're going to get from this. They assume that because it's plant-based, it's healthier. And all of those assumptions that are made are incorrect. So it's still a fat. you're still going to get fat from eating trans fats. So you're still going to be able to break this down for nutrient production. It can still be broken down and stored in the body. You're going to metabolize it just like you metabolize any other fats. So it's not necessarily terrible for you, but it's not necessarily good for you either. So that's kind of the deal with the trans fats. But saturated versus unsaturated. Saturated, again, they pack solid at room temperature, and they're typically coming from animals, while unsaturated come from plants, and they're going to be more liquid at room temperature. Essential fatty acids. So there are some fatty acids that are required in the diet. You do not have the capability of producing them. If you don't eat them in the diet, you won't be able to produce essential, like steroid-based hormones or certain types of fats that are necessary for processes in the body. So you have to consume these in the diet. A person that actively seeks out a zero-fat diet would be missing out on getting these essential fatty acids. So really bad idea to do that. So omega-3 and omega-6 are the essential fatty acids. So how does the body process fats? We're going to talk about this in more detail once we get to the digestive system. But fats are unique in that they can't just be dissolved into the bloodstream. So if we have our digestive tract here, and we've brought food into the digestive tract from the mouth, and now we're in the small intestine here. Well, in the small intestine wall is made up of individual cells. And it's the goal of these cells to absorb nutrients into the bloodstream. So if we have blood vessels here, we want to get the nutrients from the digestive tract through the cells and into the bloodstream so that now those nutrients can be transported to wherever they're needed in the body. Predominantly, they're going to go to the liver. But again, the problem with these fats is that they don't dissolve in the bloodstream. So if you do dissolve them in the digestive tract, they won't just readily float through your blood. We have to have a special way to transport them. We also have to have a special way to digest them. Because you know that if you have any like oil mixed with water and you mix them up, they're going to mix together. But if you let it stand for any period of time, those fats are going to separate away from the water. Think about salad dressing. If you get like a balsamic vinaigrette or something like that, you mix it up and you can have it all mixed in solution. But as soon as you let the bottle sit on the counter, you're going to see the layers develop. The fat layer will be on top, and like the water-based or alcohol-based liquid layer is going to be on the bottom. So inside of the digestive tract, we bring in exogenous fats. Exogenous means that it's coming from outside of the body, so we're eating the fat. So the fat's now coming into the body. Now we've got this in the GI tract. And I'll draw another side over here so it looks a little bit more uniform. Again, all these lines over here, those are just cells that line the digestive tract. So inside of here, we have this enzyme called lipase that's going to digest the fats. But then once those bonds are broken, the fats want to come back together. We have to prevent them from coming back together. So how we do that is by having bile salts surround each one of the broken down fat globules. This prevents those fats from coming back together, and this is why we need bile, and we'll talk more about that once we get to the digestive system. But the first thing we do is we break it down, and then we're going to bring it into an intestinal cell and form a micelle. So what a micelle is, it's a combination of protein and lipids to help transport the fats through the intestinal cell. So once we've done that, and now we've gotten that micelle into the cell, this intestinal cell has to process the fats that were inside of the micelle. And they have to turn them into these things called chylomicrons in order to be transported to the liver for processing. So now it enters into the intestinal cell as a micelle, and it leaves as a chylomicron. The chylomicrons go into the bloodstream, and those are then going to be transported to the liver, for further processing. Here you can see the major structure of a chylomicron. The triglycerides are trapped inside. We've got these hydrophilic heads, these proteins that are here that help support the development of this chylomicron, and now this thing is ready to be transported to the liver. Now in the liver, we call them endogenous lipids. So endogenous lipids are coming from within the body. They're either coming from the liver, from our fat cells, from any storage place that you would have these triglycerides stored inside the body. So the liver is going to do the processing of the fats. They're going to either transport the fats to your adipocytes for storage, they're going to process them for energy production, for hormone production, or they're going to transport them wherever they're needed in the body. But the way that we transport triglycerides away from the liver is by using VLDLs. So VLDL stands for very low density lipoprotein, and these primarily transport triglycerides. So just as a review, we ate fats, we broke it apart, and to make sure that it doesn't come back together, we surrounded it with some bile. Now we've formed a micelle, and the micelle is going to travel through this intestinal cell, and in the process it's going to be combined into a chylomicron. The chylomicrons can then be transported to the liver, and once they're in the liver, they're further processed to be transported by VLDLs to the various parts of where they're needed in the body. So they're either going to be stored in a fat cell or taken to a cell for hormone production or energy production or so forth. So that's how triglycerides are kind of processed in the body. Now, how are cholesterol molecules processed in the body? That's where we have LDL and HDL. So many of you have heard about cholesterol before and how cholesterol is terrible for you, and you may even have heard that LDL is good cholesterol and HDL is, or sorry, vice versa. LDL is bad cholesterol and HDL is good cholesterol. So here's the deal with the cholesterol. This is what cholesterol looks like. You should be able to identify the structure of cholesterol, and what you want to look for cholesterol is these 4 rings, 1, 2, 3, 4. Once you see the 4 rings, it's a sterile-based compound. And then these methyl groups up here, and this chain kind of signifies that it's cholesterol. Cholesterol isn't all bad. Cholesterol has a purpose in the body, and the purpose of cholesterol is to make your cell membranes a little bit more rigid. We'll talk about the structure of the cell membrane and the phospholipid bilayer and the role that cholesterol plays in that cell membrane. But for now, you can just make a note that cholesterol increases the strength or the structure of the cell membrane. In addition to that, cholesterol is the basis of your steroid-based hormones. And if you're going to process like vitamin D and be able to use calcium properly in the body, you need cholesterol. So cholesterol has a lot of very important functions in the body. The problem is there's a delicate balance between LDL and HDL. LDL stands for low density lipoprotein. Lipoprotein. Part lipid, part protein. This is a cholesterol transporter. So the whole molecule isn't cholesterol. It's transporting cholesterol. And what makes LDL bad is that this transports cholesterol from the liver to the tissues. Now, if you're consuming excess cholesterol in the diet, you're going to have a lot of LDL because it's transporting that excess cholesterol from its source to your tissues. The cholesterol can then start to accumulate inside of blood vessels and can lead to the development of atherosclerosis, and you can end up getting, cardiovascular issues from having too much LDL. HDL is considered good, and it stands for high density lipoprotein. high-density lipoprotein is also a cholesterol transporter, but this one transports cholesterol back to the liver to be broken down. The more HDL you have, the easier it's going to be for you to clear cholesterol from the body. So HDL helps get rid of the cholesterol in the body by transporting excess cholesterol back to the liver. LDL transports cholesterol from the liver to your tissues. HDL is good, LDL is bad. Now we do need some cholesterol being transported to the tissues, so we don't want to eliminate LDL altogether. But it's when we have increasing levels of LDL that it becomes an issue. Now here's a phospholipid. A phospholipid has a phosphate head and two lipid tails. So this is a picture of the phosphate head, and then you can see the two lipid tails here. The function of a phospholipid is it helps make up the phospholipid bilayer, also known as the cell membrane. And we'll talk about the cell membrane structure in just a little bit. But here's a picture showing the phosphate heads all coming together and the lipid tails in the middle. Now the phospholipid is a bilayer, so the plasma membrane, the cell membrane, is a bilayer, meaning that there's two layers of these. So the fatty tails come together in the center, while the phosphate heads form the outside of the membrane. So now I'm going to talk a little bit about proteins. The building blocks of proteins are amino acids, and this is a picture of the 20 major amino acids that help form proteins. These amino acids have derivatives, and those derivatives also make up proteins, but these are kind of the 20 main amino acids. These are the starting points for protein production. So talking about protein structure, we have primary, secondary, tertiary, and quaternary structure. The primary structure of a protein is just talking about the sequence of amino acids. So we have methionine, valine, let's see, what else can we put in here? I'm trying to find one with a small enough chain that I can draw. We'll try glycine. So that's your sequence of amino acids. It's just a chain that shows the amino acids in a particular order. Now, if you recall earlier, I talked about the basic structure of the amino acid, and we had the NH2 amine group and this COOH carboxyl group. And we had our CH. So we would have our side group here and a side group here. Now, methionine has its own side group here. Valine would have a side group here. And if we continue on the chain here, you can see how we would have this chain continuing. In order to form secondary structure is a two-dimensional folding of this chain of amino acids. And it forms in this two-dimensional way due to the side groups that are sticking out here. So as I discussed previously, all amino acids have this basic structure of an amine group, a carboxyl group, and then they all have unique side chains. So glycine's side chain is just a hydrogen. Alanine's side chain is just a methane group, or a methyl group, sorry. Valine has two methyl groups and then the CH here. So you can see the side chains that exist. It's those side chains that help to form the secondary structure of a protein. So secondary structure is the two-dimensional folding of the polypeptide chain or the protein chain of amino acids. We have an alpha helix or a beta sheet. Tertiary structure. Tertiary structure is the three-dimensional folding of a protein. So now we have a lot of hydrophobic interactions, ionic interactions, and so forth that are going to help pull this into a three-dimensional ball. Quaternary structure is the complete functional protein structure. So now we have multiple tertiary structures coming together to create a functional protein. So we have primary structure, it's just the sequence of amino acids. Secondary structure is a two-dimensional folding, which can either be an alpha helix or a beta sheet. Then we have tertiary structure, which is a three-dimensional folding. And then finally, we put multiple tertiary structures together to form a quaternary structure. Why is this important? Well, when you start to learn about oxygen binding to hemoglobin, this is a molecule of hemoglobin. It's important to know that it's actually made up of 4 tertiary structures. And within the center of each tertiary structure, you have this iron that can bind oxygen. And through cooperative binding, we can bind oxygen and release oxygen from a molecule of hemoglobin. So that's something that you will explore in more detail a little bit later on in this class, and then eventually once you get to advanced A&P. So why are proteins so important? Well, proteins help form the basis of all of the structures inside of your body. Your hair is all protein. Many of the components of your skin are proteins. So all of that keratin that fills up the dead skin cells on the outside surface layer of your body, that's keratin, that's protein. Your melanin that helps make your pigment what you look like, that's all protein. But in addition to forming the structural components of your body, proteins serve the function as being enzymes. So an enzyme is a substance that increases the rate of a reaction without being affected by the reaction. Enzymes do stuff. So sometimes you can think about them as little mouths. They're going to break apart substances in the body. But they can also build substances. They can put things together. But they're big protein complexes, and those protein complexes are going to help modify what is happening inside of the cell. So they do functions for the cell without being broken down or affected by what they're doing. The substrate is the substance that is affected by the enzyme. So if we have something like lipids, so lipids are fats, if we want to break down those lipids, we have an enzyme called lipase. Lipase is going to insert water to break the bonds of the lipids to break them down into smaller pieces. So the substrate would be the lipid, the enzyme would be lipase. Cofactor and coenzyme. A cofactor is an inorganic molecule that assists the enzyme's function. So cofactor is an inorganic molecule that assists the enzyme's function. If you don't have a cofactor, the enzyme would not be functional. So the cofactor is necessary. One of the major cofactors inside of the body is magnesium. If you don't have magnesium stuck to the surface of the enzyme won't work. A coenzyme is an organic molecule. So now it's bigger, it contains carbon, it's usually a pretty large substance that has to stick to the surface of the enzyme in order for the enzyme to be functional. If your coenzyme is missing, that enzyme will never work. So by adding and deleting cofactors and coenzymes, we can kind of turn these enzymes on and off. If we don't have fats present, why would we be using the enzymes that digest fats? Well, we wouldn't want to, so we want to be able to turn them off. Enzymes also function at a particular body temperature and a particular pH range. If the temperature shifts or if the pH shifts, it can cause denaturation of the enzyme. Denaturation is unfolding of the enzyme. So if the enzyme is denatured, it's unfolded and it's no longer functional. This is why our body temperature is maintained at 98.6 degrees Fahrenheit or 37 degrees Celsius. This is why the pH of the blood is 7.35 to 7.45. Now, this doesn't mean that all enzymes are going to function at that pH. Enzymes inside of the digestive tract may require a different pH. So, inside of the stomach, digestive enzymes like pepsinogen require the low pH of hydrochloric acid to be converted to pepsin, and pepsin's only functional at a very low pH. In order to shut off those enzymes, you dump a whole bunch of sodium bicarbonate at it, and that's going to increase the pH in the small intestine, effectively shutting off those enzymes as it goes into the small intestine. So the takeaway from this is proteins make up enzymes, or enzymes are made up of proteins. Substrates are the substances that are modified by the enzyme. Cofactors and coenzymes help aid in the function of the enzyme. A cofactor is inorganic, while a coenzyme is organic. Enzymes can be shut off by taking away their cofactors or coenzymes, and they can also be destroyed by denaturing them. And the way you would denature an enzyme is by changing up the body temperature or changing up the pH of the body. Enzymes in different locations, in different cellular locations in the body or different organs, function at different pHs. But the pH of the blood is 7.35 to 7.45 because that's the pH that the cells inside of the bloodstream are going to require in order for their enzymatic function. So enzymes are very sensitive to temperature and pH. Nucleic acids. So nucleic acids are the building blocks of DNA and RNA. So they're located either in the nucleus or floating around in the cells. Sometimes they're associated with ribosomes. And their function, well, DNA stores all of the genetic material in an organism, while RNA is more directly associated with protein production. And we'll get into that in a little bit more detail later on, but this is the basic structure of nucleic acid. So the three major parts include the phosphate group, the sugar. For DNA, the sugar is deoxyribose. For RNA, it's ribose. And then you have a nitrogenous base. There are 4 bases that make up DNA, and there's a slightly different base that makes up RNA. So for the bases, you have adenine, guanine, cytosine, and thymine. But in RNA, instead of thymine, it's uracil. Adenine is going to pair with thymine or uracil, while guanine pairs with cytosine. We'll get into a little bit more detail about the structure of DNA a little bit later in this lecture, but now you know the 3 major parts of a nucleic acid.